Okay,
The
great.
doctor can hear you.
So the class is until 9, huh?
Yeah.
That's very long, right?
Finish earlier, doctor.
Yes.
Yes, doctor, it's
I
too
hope we
long,
can
but you
end
can make
earlier.
it short.
Right?
Yeah.
We should finish earlier. Okay.
I'm hungry.
I will try to finish earlier. I will provide you all with the slides and also some of the write-up I prepare, because for this chapter, RNA-based diagnostic, I'm planning to write an ebook. So the draft is with me, so I can share that draft with you so that you can do some revision lah. Okay. So not you won't only have the slides, but you also will have a draft of my ebook content. Not published yet because it's still in a draft form, but it's something, the content is almost similar to what's in the slide. So now RNA-based diagnostics. I'm sure previously Dr. Asia already covered the DNA-based diagnostics, right? So in that DNA-based diagnostics, may I know what are the technologies or at least application that you have learned from Dr. Aziaz? Can anybody tell me?
I think one of them, it was PCR,
Mm-hmm.
polymer chain reaction, and the second one, it was isothermal amplification method, technology based.
The second one is what?
Isothermal application amplification technology.
Okay. Okay. So for that, the template is DNA based, right? Genomic.
Yes, doctor.
Yeah. For this one, the template of your target is in the form of RNA. So the technology that I'm going to cover in this topic is basically a technology to measure RNA or to detect RNA. So this class is supposed to be splitted with Dr. Eugene. So Dr. Eugene already done for his part?
Yes, doctor. We had RNA-based diagnostic with Dr. Eugene.
Dr. Eugene. So what may I know what subtopic did Dr. Eugene cover? MicroRNA? Long non-coding?
Yeah, mostly non-coding RNA.
Mostly non-coding RNA. Okay. So the specific outcomes for this topic today is to demonstrate the technology and then to discuss prospects and limitation of the technologies. It should be very short, you're right, because it's already being splitted into two. Whilst for Dr. Azia, she's covering everything herself. Nothing wrong with my slide over here. So basically, I always like to, how do I say, start the topic that with my students on the history of everything. The history about when you learn, when you discuss about, detection, measurement technology, it should go way back. Should be written in the history how the technology, what's the first generation. It's just like, iPhone. When was the first generation? What it's capable of. So similar to biomedical technologies, I think the first PCR the creation of that Kerry Mullis, I think the person, he won Nobel Prize for the his invention, or his it's his invention on the PCR. It was around 1980s, 1983 year. I think he won a Nobel Prize. He passed away, I think, last year or last 2 years. So at that time, I think the brand of the machine was Perkin Elmer, if I'm not mistaken. And at that time, the PCR was invented to measure, to amplify genomic, of course, but there was something, another discovery that happened, that that finally now, when that was discovered, we can measure RNA. Okay. Okay. So this is just basically to say that the differences between DNA and RNA, I think you have learned this anywhere, somewhere in the program, in the course. So RNA is basically more unstable compared to DNA because of the I think something to do with the hydroxy backbone or something like that. Can anybody tell me why RNA is unstable compared to DNA? Remind me again.
It's easily get degraded because there's a lot of RNAs in the environment.
One of it is the RNAs in the environment, and then good, and then another.
I guess it's sensitive to temperature.
Sensitive to temperature. That's correct as well. I think another one would be the chemical structure of it.
Yeah, it's about the structure of RNA, but I forget about this specific structure that's gonna like let RNA to be degraded easily.
Exactly. Yeah, exactly. It's something to do with the structure of the RNA. So anyway, I think all those you know, with the disadvantages of measuring RNA as a biomarker. One of it is because RNA is easily degradable. So another thing that I want to share with you is that probably many of you don't know that the DNA was first discovered by who's the person? The two guy, the famous one? Watson and Crick.
Watson and Crick.
Yeah, Watson and Crick. So he discovered DNA, and then at the same year, actually, I think in the history, if you read, only within weeks, there's another guy named Jacob something. He discovered mRNA. So within the same year, DNA and mRNA was actually discovered. I think if I open so it's in 1961, DNA and mRNA were discovered on the same year, and actually within few weeks from each other. But then DNA became the focus biomarker in understanding human disease research. And this is mostly because RNA is, the nature of RNA itself is unstable. And then so basically it's the instability of the RNA that hindered RNA to be as famous as DNA as a biomarker or as a focus to understand human disease. But so coming back to RNA, I think Dr. Eugene already covered this. There are plenty type of RNA. You have mRNA as the main messenger RNA that encoded the protein. Hold on, Okay, so mami ada kelas. You keluar dulu. Nanti kita boleh lepas ni. Sekarang ke? Ada orang tunggu ke? Hold on, class. Hold on, my children. Okay, so these slides is basically just a recap. I think you've seen this in Dr. Eugene's or maybe in other classes. There are many types of RNA. We have messenger RNA as the main, the important one where it encodes for our protein, and then there is a non-coding RNA. And the one which is the non-coding is much a much larger group of that consists of the many type of the non-coding, and these are the many type of it. We have the housekeeping tRNA, RNA ribosomal, and then we have the regulatory type, and basically it's long non-coding, small non-coding, and for the small non-coding we have several other type. So I think you can basically read this on your own. Next, and even though we have this many type of RNA, bear in mind that mRNA can also undergo this special process called gene splicing. So if you remember, RNA, after it's the transcription process, it will undergo the maturation. So basically the maturation of the maturation of RNA to become mRNA involve the process of what we call gene splicing. So RNA, when it first transcribed, all the intron exon is being read. Okay, so what the process is all about is to splice out the intron section, leaving only the exon. So the exon will be when the intron is out, the exon pull together. So exon is the one being to encode the protein. So what happened is during the alternative splicing, this splicing, we don't know. It could be random, or it could be there's some kind of signal to control the splicing. From this splicing, there will give rise to several type of variant. So it's almost like when you have mutation, you call variant as well, right? So when after mRNA, if there is different of the different splice, the splicing, it's not gone wrong. It's a normal process. This splicing can give rise to several splice variant. So mutation give rise to variant, and if splicing, we call it splice variant. Okay, so this splice variant, one example of the process is what I can relate to my job is to my research is when you have fetal and also adult form of genes. So in the fetal form, the splicing occurs. There are so many genes in the fetal form, they are different, and then the transition becoming adult, they change to something else. So for example, there is, in my work, we have this ion channel voltage gated sodium channels. There are 10 families of sodium channels. Oh, I forgot to press record. Okay. So example of this splicing is, so there are 10 types of ion channels. The one that I'm interested in is NAV, we call it sodium channel 1.5. So this ion channel, if it's in adult form, there is a special sequence for it. And then in the fetal form, there is another special sequence because of the splicing. There's some part of intron still in there. Because this intron, they are not always like splice at the same time. So apparently, in the developmental form, this splicing could occur when there is a different phase of development. So I think when you learn developmental biology, this is something which is quite important for you to learn the splicing of the genes. And it doesn't occur once, it occurs according to phases of that developmental stage. Okay. So yeah, we have all these type of RNA, but bear in mind, mRNA can also be in several type of splice variant. Okay, so because of these differences in the type of mRNA, this is actually another obstacle for RNA to become a good biomarker for detection of disease. Okay, because we are talking about diagnostic here. So because there are many types, how do we differentiate between all this? You know, from non-coding, from a microRNA, and then you have splice variant. And how do we differentiate all this? Okay. So so this topic is basically RNA-based diagnostic is how do you detect this RNA if it's so unstable, if there are so many type of RNA, how do we actually detect it? There are several. So now I'm going to go through the evolution of how to detect. Okay. So these are how we say that if you want to measure RNA. So basically, all this, the one that I listed here, like qubit fluorometer, bioanalyzer, spectrophotometer, and whatnot here, is machine or equipment or technologies that detect RNA non-specifically. So we use this machine or equipment or this technology. So basically, not for detection, but most of the time we use this technology to QC the RNA, the quality of the RNA. So qubit, spectrophotometer, fluorometer and spectrophotometer, these are direct measurement of RNA. And now these strategies are only be used to QC the RNA. How do I say, the quality of the RNA, the integrity of the RNA. If you could remember, when you measure RNA, I should have put the gel image, right? The gel image of RNA. Can you imagine the gel image of RNA? If you run RNA on the gel, can anybody tell me what do you see when you run total RNA on a gel?
You can see two bands, the 16S and the 18S.
Exactly. So you have two bands, and sometimes if your extraction is very good, you can see at the bottom very slight faint, That's what we call as I forgot at the bottom. 16. Yeah. But that's depend on the quality of your extraction. Sometimes you see it, sometimes you see like there's a smear at the bottom, and then you have the very two band. One of it is like a double the intensity is double compared to the other one. Which one is double? Should be much more intense compared to the other one?
Of course, 28. 28
Yeah.
S.
Yeah. Good. I should have put the image of RNA here. Did Dr. Eugene show it?
No, it's okay. Dr. Venu taught us in
Okay,
the
great.
UPCR report.
Okay. So, so you have so many type of RNA, and then it's unstable. And how do we actually measure or detect RNA? So the first thing before you do detection or measurement, I think in whatever you do, especially in detection and measurement, you need to create a tool that could bind to your target. Okay, so this tool, you probably think like this tool is basically like if you are measuring a protein, what are the tools that you always use? What are the common tools used for protein measurement? Can anybody tell me?
And
Uh,
I guess
mass spectrometry.
That's the machine. The
Oh.
tool in the apostrophe. Apostrophe, not is the apostrophe the
the antigen kit.
the antibody.
Ah,
Yeah, antibody.
Yeah,
Okay.
antibody. So for protein, you have antibody that could specifically go to that protein. So commonly for protein, you have antibody. So antibody here is what we call as tool, but tool in a close bracket, or what do we call that? Yeah, with that. So antibodies for protein, but what about DNA or RNA? What about DNA? When Dr. Aziah teach you DNA, what are the tools that could bind to DNA for you?
fluorescent dye or dye.
So so that labeling, so that fluorescence dye is actually what we call labeling tool. What is being labeled to the tool? Sorry, what are the tools that being labeled with that dye so that it can detect DNA or RNA? Well, in our case, RNA lah. In
Nitrogen.
DNA,
Nitrogen bases
nitrogen bases,
of DNA and RNA.
DNA or nitrogen, or well, basically it's nucleotide.
Yeah, nucleotide.
Nucleotides, okay? So you have primers. So these primers are nucleotide sequence, which is a complement to the target, and then therefore it can bind to it. Remember the primers? So in a DNA, these primers can be labeled with whether fluorescence dye usually it's an intercalating dye. So, so this nucleotide in DNA we call it primer. Okay, so so basically these are the tools that could bind to RNA. We use oligonucleotide or oligonucleotide. So there are two types. There are probe and also primer. So you know primer for PCR, right? So they are oligonucleotides. The sequence is complement to your target sequence, somewhere in your target sequence, right? Remember? So
Yes.
So it is similar to RNA. So for RNA, one of the not ancient lah, one of the classic way for us to use to detect or to measure, well, to detect first lah. It can be oligonucleotide. So it can be a probe or primer. And then now you ask, what is the difference between probe and primer? So probe is a fragment of DNA or RNA used to detect the presence of a specific DNA fragment within a sample. And then primer is a short strand of DNA or RNA that serve as the starting point for DNA synthesis. So a primer is something that when it binds, it amplifies. A probe is something that it's bind and then it stay there. It doesn't need to be something to be amplified for. Okay, so that's the difference between probe and primer. Primer as a starting point for amplification of DNA. Probe, it binds to the complement sequence and it stick there. And now what you need to do is you need to label that probe with a fluorescence or something for you to detect. If it's bind, the fluorescence produce a signal. Okay? So I don't know whether Dr. Azia covered how, but I'm sure maybe Dr. Venu or somebody. How?
Doctor, isn't
Yeah.
Doctor, isn't the different probes have different probes chemistry, like on how they give signals?
Yeah, so that depends on what labeling you use. So if it's just a normal probe, so how do I say this?
Because like for TechMan, for example, like that day that Dr. Venu taught us is that it has this probe and a quencher, if I'm not mistaken.
Yeah. So the quencher is on the other side. So because it's primer, one it's bind, and then it will initiate the amplification. And then once when it reach to the point where it met, the quencher now will be cut, and then the quencher cut and then remove, and now the fluorescence become fluoresce. Something like that, right?
Yeah, but he said that day I asked him whether the other probes from the other brands has the same working principle or not, but he said it's different.
Well, what probe are we talking about? Because the chemistry of probe, it's they call that one as probe as well. But so that probe, primer probe. So there's a combination of primer probe. I think.
Doctor, yeah, one of my readings says that the probe, like what Leo was asking, apart from the quenching effect, some probes needs a trigger to trigger the fluorescence, right? Trigger to initiate the reaction.
Trigger, they call it trigger.
Trigger, yeah. To trigger the chemical reaction so that the fluorescence label will emit the light.
Because I thought the probe, TechMan probe, they call it probe as well, but actually it's a primer. One of the primer, it has both quencher and also the fluorescence.
Yeah, I mean the other type of probe, like Leo was asking.
The other type of probe.
Yeah, the different brand of probe that have different chemistry on how to initiate
molecular beacon or something like that.
Yes, that's what I meant.
Okay. So coming back here, what I'm going to share with you here is something like a probe and primer. The difference between the two. The probe is a fragment of DNA. It could bind and detect the presence of specific DNA fragment within the sample, but it doesn't serve as a starting point for DNA synthesis or amplification. Okay, and then the primer is something similar, but then it should be in a pair form, and then it it will initiate a starting point as for DNA synthesis. But anyway, I should have explained better, kan? I mean, how do you define all this? But anyway, maybe when we go to the technology, you will. It becomes confusing now, kan? The name of the probe and the primer, because in the
Yes, it is. I think
Is it primer means elongation will happen, whereas probe
Yeah.
there's no elongation.
amplification will happen. Whilst the probe amplification does not happen, you just bind, it will bind to its complement, and then you just need to modify the probe so that when it's bind, it produce a signal.
So both the DNA and RNA we can use oligonucleotide.
Yes. So if you are measuring RNA directly, so you can design your probe. You can design your probe using either DNA or either RNA. I think vice versa lah, depends on the sequence. If it's RNA, you have just to replace the, what do you call that? Uracil, right?
U to T.
Yeah. Okay, the other differences used to detect a specific DNA fragment in qPCR, and then used to initiate the DNA replication and initiate PCR. So see, in the second line, you see there, used to detect specific DNA fragment in qPCR. So basically, in the real-time PCR or qPCR, it is a combination of primer and probe. So the one part is the primer to initiate, and then the other is a probe that has quencher and also the fluorescence together. So that's how the TACMan works. Leo said here, but I thought you can only use qPCR to quantify RNA. You can use qPCR to quantify RNA and also DNA. You can use both because qPCR is quantitative.
Doctor, what is RNA probe? Is there something that's called RNA probe?
RNA probe is the sequence contain uracil.
Oh, okay.
You know, everything is similar except for that particular nucleotide is different.
Because I thought in the normal qPCR workflow is that you have to convert the RNA into a cDNA first, and then you use a DNA probe to.
Yeah. If you convert now, if the technology you are using, you are converting your RNA to DNA, then you are using you can use DNA primers or probe, primer probe combination. But there are technology, for example, northern blotting. Have you heard of northern blotting? And
northern blot.
Yeah, northern blot. So in northern blot, you are measuring RNA. You don't convert it to DNA. Okay? You are measuring the RNA as it is in the sample. So you need a probe or a tool that could bind to your RNA.
I see. Okay.
Okay? So, okay, now the length there, the differences of the length, and then it said there hybridized with double-strand DNA, whilst that one hybridized with single-strand DNA, the primer. And then probe generally labeled with a fluorophore for detection, and then primer can be labeled based on the purpose. Okay, sometimes in qPCR, you don't have to label your primer. You use a naked primer. You add a dye that could detect double strand DNA, a product from that from that qPCR. Okay. So this is a new tool that recently be found that could bind to RNA, and you can use it for detection other than the DNA or RNA probe and primer. And that tool is called guide RNA. I think this is especially become famous after the CRISPR being, the CRISPR, the gene editing thing being found. So in that CRISPR technique or method, or I wouldn't say it as a technology, you have an enzyme that called Cas, right? It can be Cas13, Cas whatsoever, but there is another part that called a guide RNA. So this guide RNA is the one that will go inside the sample and detect your target, and then once it there, the Cas will go and do its job, whether it's to edit or to do its job lah. The Cas enzyme. So Cas is actually the enzyme, but the one for detection is this guide RNA. It's almost similar to the primer and probe. It's a sequence of something that complement to your RNA. It's just that it has this particular, what do you call, chemical or another sequence that can be for a specific Cas to come in. Okay, so this guide RNA is a specific RNA sequence that recognizes target RNA or DNA region of interest and directs the Cas nuclease there for editing. So for RNA, for gene, do you remember CRISPR-Cas, what the name of the Cas for gene editing? Genomic editing.
Cas9. Cas9.
Exactly. So Cas9. So for RNA, it's called Cas. Have it here. Cas13. Okay, Cas13 and also Cas13. Yeah. Hold on. My son must. Okay. So there is these three tools now emerge as the tool that could bind and can be used to detect and measure RNA. Probe can be probe, primer, and now this new one called guide RNA. So basically, is there anybody covering this CRISPR, how CRISPR works in the lecture?
No, not yet.
Not yet, huh? I think semester 2. Semester 2, 511. No, not 511. 510. You have 510 in semester 2, right? Forgot. You have 529 and then 510. But anyway, so for RNA-based diagnostics, you have your RNA, and then you need to have a tool that could bind and to be used for detection and measurement. We have probe that could bind to RNA, we have primers that could bind to RNA, and then we have this new one, Oh yeah, second semester, it depends, industrial or research, right? So if you choose industrial, maybe um you won't. But nobody in the first semester cover this CRISPR, how CRISPR works, huh? No, okay. But anyway, so basically this detection using guide RNA, it's not for editing. You are not using this guide RNA CRISPR Cas13 for editing the RNA or whatnot. You just use it for detection. It's basically for diagnostic detection. Okay, so the guide RNA is just like the probe and primer. I'm totally blur here. Kavita, tell me, which one did you blur?
But actually, I have no idea about this CRISPR. So CAS is very new for me because I never. I mean, this semester I only have bioinformatics and this course. So I'm not sure my other batchmates have gone through this in other lectures. So for me, this is my first exposure.
Okay, I think maybe I can suggest Dr. You know what? Anybody in the workshop choose something diagnostic using CAS? Have you
Yes,
choose your
doctor. Like, I choose it CRISPR as my topic for workshop. So CRISPR, it's all that I covered. It's all about genetic engineering, like how guided RNA guiding the CRISPR Cas9 protein to cut the DNA, the specific DNA of the gene, and replace it with a new one. The replacement is undergo the DNA repair system within the cells. So this
That's
is
for the
all,
editing.
yeah, for editing, but not like a probe. This is new for me. Like, I thought like CRISPR just use it for like gene editing and cutting, scissoring, and that's it. But right now I see like it's just probe thing detection.
Yeah, it can be used for detection diagnostic. It's not for editing the gene or whatnot, but it's for to be used as a tool that could bind to your target, and then you need to modify that guide RNA with certain labeling, so that
Yes.
after the cut, it will produce signal.
This is the cutting. It's all coming from the Cas9 with the guided RNA. That there is like different portion of the protein that finding there's a PAM, like finding the specific targeted sequence in the DNA or RNA. So then after binding to the target sequencing, and then the guided RNA will be complementary to the sequence, the sequence of interest, and then the cutting will happen by two of the ligand of the protein. After the cutting of the DNA, the sequence of the nucleotide, and then the cells take the action for repairing it. So repairing by the end joining or like cutting. So here's like there will be check out and check in of the gene. It happened, and then the CRISPR like working like that. It's all about the Cas protein.
Yeah, it's all about Cas9. But anyway, I think maybe I can suggest to Dr. Nurul, since if some of you choose industrial pathway, you wouldn't be able learn CRISPR, because CRISPR has been taught in the second semester only for those choose research pathway. So in this sense, maybe I can ask Dr. Nurul maybe do a seminar, and then we can invite expert on CRISPR to do that seminar. Will you all be okay with that?
Yes,
Okay.
of course, like we need that thing.
Okay, it's not something like it will be an exam. I'm sure it's not an exam because it's not been covered, but at least after the seminar you will get to know CRISPR in gene editing, in which you are changing the genetic material, and then CRISPR that being used as a diagnostic, as a tool, especially on the guide RNA. How does it work for it to become a diagnostic tool for RNA detection. Okay? Okay. Again, this is some more about the the guide RNA. And this is just an additional reading for you. Okay, now in this additional reading, you can see there the word amplification free diagnostics. Okay, so for amplification free, if your detection method involve primers, or your detection, there should be amplification free and also amplification dependent. So the opposite of amplification free is amplification dependent. Yeah. What's this? Okay. So for detection, actually this amplification free, application dependent, we should have taught you much earlier during Dr. Azia's class. Where is reverse transcriptase? Guide RNA, CRISPR. Okay. So this one. So before I go further on the technology, whether it's amplification free or amplification dependent, so when the word amplification free means that you need to amplify your target, and when there's amplification involved, of course, the tool that you use is what do you call that? Primers. Okay, and it's basically polymerase chain reaction. Or yeah, but if you are using amplification-free technology, and this amplification-free meaning that you have your sample, and then you have, and most of the time, whether it's a probe, and then that probe binds and then you detect. You don't need to have, it doesn't involve amplification. It binds and then it produces signal. So this is what we call amplification free and also amplification dependent. Okay, so primers for amplification dependent, probe or that, what do you call that? What's that? The CRISPR one. Guide RNA, it's amplification free technologies. So you have your RNA target, you need to have your tool, and then next I will cover the labeling. How do you label this probe and primers? But before we go to that, I would like to, it's not introduced because I think Dr. Venu already covered the reverse transcriptase. So when you are using your RNA as your target of interest for diagnostics, you can measure your RNA directly, but because of the instability of RNA, most of the time now what we do is we convert the RNA to become a complement DNA. So when it is in a DNA form, it's stable, and then the technology wise to detect this DNA is you have an option, you have several more options. So what is reverse transcriptase? It's actually an enzyme discovered in retrovirus, and then it is actually the most dramatic scientific moment discovery of the second half 20th century, and it's basically revolutionized molecular biology. And this is in the molecular cloning part. Its result was a major paradigm shift, reversal of the central dogma of molecular biology. Central dogma, you have your DNA, RNA, and then protein. Well, anyway, you don't have something like protein become RNA. So the reverse is at the RNA now you can change it to become DNA. Okay? I'm not sure when they discover this. What year is it? So this is just to remind again, if when you due to the RNA weaknesses, it is easily degradable. And if you are measuring RNA, you are adding more cost to your diagnostic methods if you are using RNA. If you are measuring it directly using probe or whatnot, that's okay. But when it involves RNA convert to cDNA, here you are adding some more costs. If you do a break-even cost for your diagnostic preparation or your diagnostic invention, you need to include the cost of converting your RNA to cDNA, in which you use reverse transcriptase, and then you have once you and then when it's in a DNA form, and then you can use the technology for the detection. Here, just to share with you, your RNA, you can convert it to cDNA, and then if you want to keep your cDNA, you want to amplify it more and more, you can do cloning, and then you can keep your cDNA in a bacterial plasmid or in a bacteria. So you are amplifying your target, and you keep it in somewhere stored. For example, if you have a patient sample, let's say very little amount of the sample of your patient, and what you do, you don't want to use the whole RNA because you will run out of that RNA. So with the discovery of reverse transcriptase, what you can do now is you convert the RNA of your patient sample. Leo, you mentioned something. Is it possible to amplify RNA? It's not amplifying the RNA. You convert your RNA first to complement DNA, and then your DNA, you clone it into a bacterial plasmid. And then that plasmid, you transform it into bacteria. And then your bacteria, when it's dividing, meaning that you are making it more, you are amplifying your target. So your bacteria now contain a genetic material, RNA, of that patient. You understand? Anyway, so that's what it means by cDNA libraries. A library of cDNA libraries of a certain person or certain gene of that person. It's really tough to teach this. Any question that you want to raise?
Yeah, doctor was wondering if a RNA virus infects our body, it will be able to replicate.
RNA virus replicate in our body by using our transcription process, right?
Yeah.
Yeah. So through that, it amplify more of its RNA. So is that why you are confused?
Yeah, doctor, how can that RNA be amplified? Why is it not possible to do it in vitro?
Because, no, basically they have this, so this virus, well, actually the origin of reverse transcriptase is due to this RNA RNA type of virus. So once they have RNA, or basically all virus, is it? Oh my God, I'm not really an infection person. We don't basically replicate. We just learn that how they do it is by reverse. We know that this reverse transcriptase can make from RNA to DNA. And then from that information knowledge, we took it to create a method for us to understand, for us to understand or to do experiment with it. And in this case is to do the conversion of cDNA. We I think maybe Dr. Venu can explain it better because Dr. Venu is the infection virus guy. But the one that I want to highlight here is that the discovery of the reverse transcriptase from that virus, why we cannot copy that, instead we use this method. Okay, so can we stop a bit to let the azan finish and then I continue again?
Yeah, sure.
Okay, thank you. Okay, so we will continue and maybe we will stop at around 7 or maybe 8 for the praying, to allow the Muslim to pray. And then we continue after praying, maybe like 10 minutes, and then we finish off. Is that okay? I will try my best. So, do you ask this much of question during others' class?
No, Doctor, I guess
Yes, of course, we are curious students.
instigate our curiosity.
We are students like to learn.
I wonder during Dr. Azia's class, you ask this much as well.
Yes, I think so.
Okay, good, good, good.
You could ask her.
Okay, so basically, what was it when yeah. So the one that you said, why can't we replicate that what happened in the virus replication in our body, for technology? So replicate that and then do it in a technology wise. I don't know how to answer that, but what I know is that we understand viruses, especially the retroviruses with the RNA genetic material. They use this reverse transcriptase to convert their genetic material RNA to DNA. And then with that knowledge, information that we gain, we take that knowledge and then we create a flow of work in which we can keep our genetic material target sample. Let's say if it's an RNA template, we can keep that RNA template in a cDNA form. And if we want to make it more, we can change that cDNA template into a library to amplify it more in expression systems such as bacteria. Okay, so when you do have diagnostics, let's say you have sample patient, and this patient, for example, brain cancer. And in brain cancer, of course, during the biopsy, the sample being taken out is like very small, very little. And you want to measure a lot of things with that small sample. Some want to measure protein. In fact, if your diagnostic is multi-omics or transdisciplinary, multi-omics and transdisciplinary, you need to measure. Let's say you want to measure protein, you want to measure transcriptome, and then you want to measure DNA genomic. So in this sense, how are you going to leverage or divide this sample, very small sample, to measure all these omic that you want to? So one of a way strategy is, if it's genomic or transcriptomic, you can actually create a library of that sample of that particular patient in the form of library. So you clone it into an expression system. So that expression system, you can label it patient 1. So if you have a bacteria, you have many. If you want to study patient 1, so this bacteria clone contain genomic of patient 1, and this is cDNA libraries of patient 1. And then the other for protein, you can use it as a one-off. Protein, we don't keep it. Once you lyse your sample for protein, it's gone. And if your experiment is not working, it's gone. But for genomic and transcriptomic RNA, you can keep it by doing cloning. Okay, so that's what I mean when I say, for detection or whatever. So this is basically a more downstream strategy of how you want to keep your sample patient, very precious one, when it's involved, genomic or transcriptomics. Anyway, so that's basically what I can tell you. If coming back to Kavita question, I cannot answer it properly. Maybe Dr. Venu or Dr. Eugene can help out. Maybe I can pass this to them, or maybe you can ask them directly. Okay, so the libraries The difference between genomic library, DNA, and cDNA library. So genomic library is a collection of DNA fragment contain self-replicating vector, blah, 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 and it of individual DNA. And then usually it's much larger compared to cDNA library. Why? Because genomic contain both exon and intron. So we know that genomic is much larger compared to cDNA library, which is cDNA is a complement from the RNA. Okay, so RNA you reverse transcribe, making it DNA. So DNA library represent a collection of only those DNA fragments that were transcribed into mRNA in the cell from which the mRNA was isolated. So basically, these are the difference between the two. And this is just to imagine, to help you to imagine the difference between the two. So for genomic library, you extract the genomic material, you clone it into plasmid, and then you transform it into bacteria. If it's cDNA library, your sample, you extract the total RNA. And then from that total RNA, you reverse transcribe, making it to complement DNA. And then that cDNA, complement DNA, you clone it into a plasmid vector. And then when it's ready, you transform it into a bacteria. So these are the two strategy that you can use, especially when you are dealing with a precious sample. So this is very important in diagnostic because remember, precious samples very small in size, and this strategy can save your sample. You can use it infinitely as long as you have your libraries being kept properly for every patient. Okay? Now, again, this is just to highlight the differences between genomic and cDNA libraries. Now, we have in RNA-based diagnostic, you want to measure RNA. And how do you measure RNA? You have to have a tool that could bind to your target RNA. And this tool I have shared with you, there are 3 types, and these are all oligonucleotides. If it's protein, immediately common antibody. Okay, but when it comes to RNA, oligonucleotides. And these oligonucleotides that could bind to RNA, it can be as probe, it can be as primers, and the other one, we call it as guide RNA, the very latest one. Now, coming back, now you have your tool, and then did I put anywhere in the labeling here? I was looking for the labeling. Okay, the labeling will come later. So now when you have your tool ready, you have your target RNA, you can choose whether to convert to cDNA or you want to detect your RNA as it is. So this is when the amplification free or amplification dependent comes in. Let me do, so if it's amplification free, the term that usually being used is hybridization. Hybridize, bind to, hybridize, bind to, okay? Amplification free, hybridize, amplification dependent, meaning you amplify. Can I ask you, PCR real time, if it's amplification free or hybridized, amplification dependent?
Amplification dependent.
Yes, amplification dependent. Microarray?
No, it's independent.
Microarray, nanostring is considered microarray.
Nanostring is another.
Oh,
Is another. The chemistry is a bit different than microarray. So now the question, microarray, if it's is it dependent free or dependent amplification dependent or amplification free or hybridization?
Free.
hybridization.
Yeah, hybridization or amplification free, correct. NGS, RNA sequencing,
Amplification dependent.
amplification dependent. In situ hybridization,
I mean, it's hybridization itself.
hybridization itself. So it's hybridization. It's amplification free. Northern blot,
Amplification free.
amplification free or hybridization. So basically for this amplification free or hybridization technology, it uses probe. Simply you can say it uses probe. So that probe can be sequence of RNA or sequence of DNA. Okay, it hybridized to your target. Now, so after so many years of discoveries in biomedical research, biomedical technology, it is still to say PCR continues to serve as the backbone for most molecular diagnostics. In fact, even for NGS, you need to validate your NGS with real-time PCR. So you can search this even with the latest, next gene sequencing. Once you have your target, even though NGS can provide data for whether upregulate and downregulate, but if you were to validate your NGS, you need to go back to real-time PCR. Okay, so that's the gold standard for. And so real-time PCR is an amplification dependent that you can use for RNA detection. Okay. And but the sample, the template that you use, it needs to be in what form? If you use PCR or real-time PCR for detection of RNA, your sample need to be what?
Need to be
DNA?
complementary DNA.
Yeah, you need to convert it to complementary DNA first. Okay? So the first generation is PCR, qualitative. This is the first generation. And then we have the second generation, real-time PCR, in which you add a labeling for you to detect signal. So that labeling is fluorescence. It could be fluorescence dye, or it could be a specific fluorescence attached to your probe. And that combination probe work together with your primer. It's a probe primer combination, TechMan. Okay? So earlier, PCR earlier in the first version of the real-time PCR, we use ethidium bromide. Usually in the PCR qualitative, you use ethidium bromide after endpoint, right? Endpoint, once you finish your cycle, you add ethidium bromide. But in the very early invention of real-time PCR, instead of using, the fluorescence dye or whatnot, they use still the ethidium bromide inside the reaction. So the ethidium bromide will bind to double-strand DNA and produce a signal that will be captured by the camera inside the machine. But later on, when the discovery of this intercalating fluorescence dye, cyber green or or evergreen or whatnot, we replace ethidium bromide because ethidium bromide is more toxic, carcinogenic, and whatnot. So we now use intercalating dye called cybergreen or evergreen. It binds to double strand. Okay. And then we have now digital PCR. So this PCR amplification free, if you were to use it, you need to, like said, we need to convert it to cDNA complement. But remember, during the COVID time, we have a real-time PCR to measure COVID as a gold standard. I'm not sure whether you guys remember or not. Okay. And that real-time PCR of the COVID is what we call, it's a one-step PCR. Meaning that you have your total reaction in one tube and you have reverse transcriptase inside the tube. You just put your sample inside everything. And then first step, you need to change your RNA to cDNA and then inside the same tube, and then you change the temperature so that your Taq polymerase will start to run its job for the amplification. I should have put it here. The two-step and one-step PCR. Have you heard of it?
Yes,
Anybody?
yes. One step is just you're doing without converting to cDNA. Like
Yeah.
you just put it in one go and then
Yeah.
it convert by itself and then it will. Yeah.
It
Two step is
in a different tube.
in a different tube.
Yeah. So two-step
Two
means
steps that
convert cDNA in a different tube and then you add that sample from different tube inside your new tube for Taq polymerase. But one step means your reverse transcriptase is inside one tube. Everything is one tube. Reverse transcriptase is there. Taq polymerase is inside there as well. You just change the temperature. Okay. Anyway, so again, reverse transcriptase, you use template whether it's RNA or cDNA. Like I said, if it's RNA, you can use one step. If it's cDNA, you use two step. You convert it to everything in two tubes. So what you need, components of reverse transcriptase. I think, I don't know, Dr. Azia or Dr. Venu already covered this. You have to have reverse transcriptase, your target RNA, DNA polymerase, primers, your DNTPs, PCR buffer, real-time PCR chemistry, the dye, fluorescence dye, and then you have your machine, real-time PCR machine. And that machine contains a camera to capture the fluorescence signal. Okay. So primers, dye-based chemistry. So this is the labeling of the tool. So you label your primer, or it can be a probe as well, with a labeling compound. And that labeling compound, currently we have the most famous cybergreen or evergreen or the TechMan probe. But previously, before all these fluorescence color being used, what are the most famous labeling method? Can anybody guess?
sorry.
Before all this fluorescence labeling.
Just now you was mentioning ethidium bromide, right?
Ethidium bromide, again, it's fluorescence.
Oh, you mean before fluorescent?
Before ethidium bromide, before that.
Ah, it's a good questions.
Yeah.
Never thought
Before all
before.
this. How do you think researchers label the probe or the tools? Radio label, yeah, correct, Yin. You use radioisotopes to label. And usually we use it, what do you call that? Phosphate 32, P32. So that's the very common radioisotope being used to label your probe. And then you have to use radiography to measure the signal. I think now, because of a lot of people are afraid of radioisotopes. Hold on, your class. Hi students, can we stop now? Can we stop here and then we continue again maybe in around 8, 8, something like that, to allow Muslim to pray? I need to pray as well.
Yes,
Yeah,
Doctor,
all
it's
right,
gonna
sure.
be better.
Okay, and you probably can grab a quick dinner as well. Okay, see you.
All right, see you, doctor.
Can we continue? Ah, I was thinking to finish this earlier. But now it seems like it won't be as early as we promised. Anyway, so so the labeling. So once you decide your tool, you need now to decide on what label your tool need to be labeled with what. And this labeling depends on the technology, the machine that you want to use. And if it's fluorescence, you need to use a machine that could capture fluorescence signal. And the machine that capture fluorescence signal, and usually it relate to camera that catch the wavelength of that fluorescence signal of the specific wavelength of that specific fluorescence signal. So now, the real-time PCR, so your tool, if you use fluorescence dye or, fluorescence dye, you choose a technology that have a camera to capture that fluorescence signal. So PCR contain a camera that does that to capture the fluorescence signal of your detection. And plus, if it's a real-time PCR, because you are using something that measurable, it becomes real-time PCR. Anyway, so if and I also ask you what are the classic way of labeling, and Yen answers radio label. So so radio label is a very classic way of labeling the tools that could for the detection. Okay, so other than radio label, can anybody give another example of labeling? We have fluorescence, we have fluorescence or the labeling called fluorophore, and then we have radio label. Can anybody say, give me another option for labeling? Anything that come up to your mind? Biotin. So biotin is another way to label detection. Okay, so biotin. So in biotin, you need to have a chemiluminescence, chemiluminescence to capture your signal. So biotin is really chemiluminescence lah. And then there's another way you can have a luciferase, but luciferase is actually fluorescence. So you can label your tool with all these labeling method. Fluorescence is the most famous one. The old most classic is radio isotope labeling. And then you can also label it with biotin. Okay, so fluorescence or fluorophore labeling, that relate to, well, because PCR is still the most famous technology to measure nucleic acid, and RNA is one of it. So I'm going to use example of real-time PCR, especially reverse transcription real-time PCR, primers in combination with probe, as an example of technology for RNA-based diagnostics. So basically, this is what the components of reverse transcriptase real-time PCR involve. I think, like you said, Dr. Venu already covered this. So it's easier for me. You have reverse transcriptase first to convert your RNA and making it to cDNA. And then, of course, you need to have your RNA as the template, reverse transcriptase. And then you have DNA polymerase to amplify primers and DNA polymerase to do the amplification. And then you have to have the NTPs and buffers. And that is the chemistry, the fluorescence. That's the probe. And then that probe is labeled with fluorescence and quencher. And then you have another part, which is the primer. And that's why it's a combination of probe and primer. So once it's amplified, and then a part of that primer, when it's meet, it will cut the quencher. So when the quencher remove, the fluorescence now fluoresce. Okay? So the quencher and the fluorescence, the probe, are being labeled on the same probe. They need to stay together so that the quench, will absorb. Quench means absorb the fluorescent signal. And when the quencher is being cut off, removed, now the fluorescence can produce the signal to be captured by the camera inside the machine lah. So basically, these are the components for reverse transcriptase real-time PCR in RNA-based diagnostics. And then these are basically how the primer probes works in the real-time PCR. You have primer there, you have probe, and then once it meet, it will cut the quencher off, and now the signal will fluoresce. So these are the chemistry for the TechMan. I know when you said there are several chemistry, there are molecular beacon and whatnot, and every type of that works in a different way. But the one that I'm highlighting here is primers combination with probe, and that probe is labeled with this quencher and fluorescence. Fluorescence label. This is again, this image is to give a difference between one step and also two step. And you realize here that for one step, you have one tube contain everything. You have your reverse and you have your polymerase, DNA polymerase inside there. But if it's two step, you have to convert your cDNA in a different tube. So you have your cDNA tube, and then once that cDNA ready, you transfer it into the reaction mixture tube to be put inside the machine to run the PCR. So that's the difference between one step and two step real-time PCR, reverse transcriptase real-time PCR. Okay? Because if you say real-time PCR without the front reverse transcriptase, that real-time PCR can measure both. The template can be DNA. Okay, so real-time PCR can be used to measure genomic as well. But if you put reverse RT PCR, reverse transcriptase real-time PCR, your template is actually cDNA or the transcript origin of the sample. Okay. So this is just to show, I think Dr. Azia also have shown you this. This is the steps. So you have different temperature. So that temperature is actually to activate, to denaturate the double strand, and then you deactivate the enzyme, and then you start the reaction. And then it cycle again. Everything is being cycled again. So this is how it looks like. And then when in the real-time PCR, when it says real time, the signal of the fluorescence is being captured, and that capture of signal is being turned into a digital number, which the fluorescence intensity at that particular wavelength. Now it has changed into a digital number, so you can trace how much of the product being amplified. Okay, so for every cycle, it will start to amplify, so meaning more fluorescence is being captured, and then after some times, all the components run out. You run out of nucleotide, the NTP. You run out of primers, and that's why it starts to plateau over here. Well, similar to bacterial growth or yeast growth, when the nutrient finish, the growth start to plateau. So similar to this reaction, once all the reagent, the component finish, the production of the product start to plateau. Okay, so this is what we call amplification curve of a real-time PCR.
Doctor, the CT or CP, is it CP or CQ?
So the classic, not long time ago lah. The classic name is cycle threshold. CP here, the point of meeting between threshold and your amplification plot. CP. But now they change CT to become CQ. Q means quantified cycle or something. Cycle quantified.
Okay.
So you can double check that lah. But CT equal to CQ now. CT is the old name. CQ is the new name.
Okay.
I think most of the machine now, they have set the machine with the CQ, because just like the housekeeping gene. The old name is housekeeping, but now they want us to use reference gene instead of housekeeping gene. But both are similar. Okay, so CT, the old name, CQ is the new name. Okay, so in the machine, this threshold line, this purple or this blue line, you can move manually this blue line. So once you have your machine, you can use the setting in the machine to move this threshold, and most of the time you can also choose to set it automatic at the machine. So threshold, you click automatic, so the machine will adjust the threshold at the automatic level. Anyway, so this threshold is important because the point where this threshold meet with your amplification, this is where your the number of detection. This is where the one that you will take as a detection number figure. Okay? Because you have your amplification along the way. And then you probably will think, so how do I choose from is it this point or this point or which point? So the machine usually, if you click automatic, the machine will do a automatic configuration, and it will set automatically where the threshold best fit should be, the best fit threshold. Okay, so the point, this CP point is where the number falls straight down at the bottom. Let's say here it says 19.8. So this is the point where it will give you the measurement. CQ point at 19.8. So Yan asks, in what case we need to choose the CT on our own? In my case, in what case? In my case, usually, we run our we run our samples together. Sorry, the gene that we run to the amplification real-time PCR, usually we run it with reference gene, beta-actin, for example. Okay, so you should have your beta-actin somewhere here. Let's say another line. Oh, where is the point? Okay. And then you run your sample, you have your beta-actin, and usually amplification, you will have two lines. Where is the pointer? So, and most of the time, it depends on your pipetting. Your line, your two target and housekeeping don't in parallel. Because housekeeping, usually they appear much earlier compared to your target gene. Let's say it appear around 10 or something, but it plateau much earlier, let's say. So you need to adjust. It depends on when you do your standard curve. Oh my God, this is a very hard question actually, Yan. In my experience, I will always set the threshold at the reference gene that usually my reference gene appear. So usually my reference gene appear at around 10. So I will adjust the threshold so that my reference gene will become 10, let's say 10. And then at that reference gene 10, I will take my target. My reference gene 10, what is the reading for my target? So that's when I do manual. But usually, if you have you run your sample together with your standard, because if you go through how you prepare standards, the your the do we have a picture of the amplification? Yeah, amplification. For example, here this one. You see the amplification? Where is my cursor? Okay. So here is the amplification. You can see when I move this? You can see it, right?
Yes, yes.
Yeah. So usually when you run your sample together with your standard curve, the size between the amplification, the size between the first one and the second one, the standard curve with the serial dilution, you will have similar size. The size between the first and the second over here, this line. Oh my God. There should be another class for this. How to add. Yeah, yeah. If you cross this line over here, the size from the first line to the second line, if you prepare a serial dilution, it should produce almost a similar size, the width. And it depends on your pipetting. Sometimes the students, when they pipette very well, you will get that very nicely separated with the same length. But if you don't get that very nice separation, this is when your automated threshold is useless. So in that case, you might opt to do manual threshold set. So that's the answer for that. But I think we need to have a separate session to explain that even more. But anyway. Okay. So when it's RNA detection using reverse transcriptase real-time PCR, so what are the applications? So you now know there is you want to detect RNA. You need to decide whether to use primer, probe, or guide RNA for your detection. But this depends on the chemistry of labeling, if it's fluorescence or radioisotope or biotin. But the most famous labeling of this tool, primer or probe, is fluorescence. Okay? So one technology that is famously used for RNA-based diagnostic is reverse transcriptase real-time PCR. Okay. And other technology can involve amplification-free. Real PCR is just amplification dependent. And there are several other technologies which can be amplification dependent or amplification free. And then I share with you already example of amplification free. So if you choose reverse transcriptase real-time PCR for your RNA-based diagnostics, and then now you ask, what are the application? So when the question is what are the application, you need to go back to what disease are you studying? Okay, or what disease are you applying this RNA-based diagnostics of reverse transcriptase real-time PCR? So in the example of disease I'm sharing here, cancer, RNA-based diagnostic in cancer. Reverse transcriptase real-time PCR can be used to amplify target genes associated to splice variant. Remember, I mentioned the RNA, the mRNA splicing, you will have several splice variant involved, and maybe you have a specific splice variant for that particular process, maybe metastasis or whatnot in that particular cancer. So you can use reverse transcriptase real-time PCR to amplify target genes associated to splice variant. Okay? And then the next application in cancer is you can use this technology to amplify predictive genes associated with certain targeted therapy. For example, you measure in cancer, you can measure estrogen receptor, or you can measure progesterone receptor. Okay, because these receptors are associated with a drug called Herceptin or tamoxifen. It's a monoclonal antibody for breast cancer. Breast cancer positive for estrogen or progesterone receptor, hormone receptors. Okay? And then the last one that I can share with you is to amplify signature genes associated with therapeutic for response, relapse, and recurrence. Okay, signature genes associated with therapeutic response, relapse, and recurrence. So these are example of application RNA-based diagnostic in cancer using reverse transcriptase real-time PCR. And RNA-based diagnostic in infectious disease, what about for infectious disease? Reverse transcriptase real-time PCR. Example here is COVID-19. So reverse transcriptase real-time PCR have been used for COVID-19 to amplify target genes associated to virus. So for COVID-19 is the RNA type of virus. So these RNA in the COVID-19 patient sample, you take the patient sample, you extract the RNA, and then you put it in the real-time reverse transcriptase one step. The one that we use in the clinic, in the lab for COVID-19 is a one-step reverse transcriptase real-time PCR to amplify target genes associated to COVID-19. Okay? And then also for infectious disease, another famous one is HIV. We use reverse transcriptase real-time PCR to do viral load assay of HIV. Because as we know, HIV, we measure viral load to check whether patient is at what stage, whether it's HIV or it's already at the AIDS phase. Okay, because not HIV patient enters AIDS phase, AID. So we use this particular assay, viral load assay, to measure that HIV in the patient serum. So these are examples of application of reverse transcriptase in real-time PCR. And then for other technology, northern blot, this is hybridization, laboratory method to detect specific RNA. So you have your RNA samples, you put it there, and then you have your probe labeled with maybe radioisotope or fluorescence. And then if you are using fluorescence, you need to have an image analyzer to capture the fluorescent signal. And if it's a radioisotope, you need to have a radiography. Usually it's a what do you call that? Just like like a photography, but you need to expose it with the different reagent. So this is northern blot hybridization. You can have a look further. Extra reading. And then this is in situ hybridization, fluorescence in situ hybridization. You have your DNA, and this or it can be cDNA. And then usually you have your probe tagged with fluorescence, and then your probe will bind to your target cDNA here, convert to cDNA, and sometimes you can use intact cells to detect mRNA in the cells as well with the in situ hybridization. So again, visualize with camera for the fluorescent signal. And then this is for the use of RNA Cas13 CRISPR-based diagnostics. So this is an example of how CRISPR-Cas13 being used for diagnostic. I think example here for what? No? They didn't mention the type of disease. But I'm sure if you have a good RNA sequence as your marker, you can definitely design the guide RNA complement specific to your target RNA, and then you can follow the step as it is now. So so this is nothing. This is not into this one. Okay, I will delete this from the reference note. But this is the last one, prospects of RNA-based diagnostics. So as we know, RNA, they are not stable, and plus for the detection. But now with the emergence of new technology, especially with the reverse transcriptase, we can design our own assay for RNA, to detect the RNA. But bear in mind, there is also, what do you call that? Drawbacks when you use RNA as there's a drawbacks, there's a pro and cons, not drawbacks all the time. There's a pro and cons. So I'm sharing with you, this is particular readings that have highlight the difference, the comparison between RNA-based diagnostics, which cannot be learned directly from genomic mutation or genomic diagnostics. This one is a very good reading. So I would suggest you have a read of this particular paper to cover our specific objective to discuss prospects and limitation of the RNA versus genomic diagnostics. So read that 2020 article and everything is there. But anyway, I don't think this year I want to create exam question on the prospects and limitations. So just have a read for your own knowledge, and then but the rest is, yeah, the exam will not covering this one. It's just for your knowledge. The exam is more on the one that we discussed much earlier. So yeah, so that will be all the topic for today. I hope I still managed to finish only half an hour earlier compared to what we promised. I have already recorded this session. I will post it to you guys. And also this slide, I will share with you all as well. And as I promised, I will share also the draft of the ebook content I'm writing about this RNA-based diagnostics. Any question?
Doctor,
Yeah.
mostly like microarray or RNA sequencing or even nanostrain is for research purpose only, not for diagnostic. Why?
Most of the research are not on diagnostic wise, huh?
I mean, mostly these applications, I mean, these platforms is for research purpose only. I mean, it's already mentioned in their technology that this is for research purpose only, not for diagnostic. Why?
Why? Well, actually, that's a very strong statement to say that you only use microarray, sorry, you use microarray or NGS for research purpose. Because these days, microarrays and NGS already being used for diagnostics. In fact, some company have designed a panel of gene to be run on microarray platform or to be run on NGS platform as diagnostics.
Oh, okay. Maybe Malaysia, we didn't use.
We didn't use because our health system is subsidized by government.
Yeah.
Okay, and only these days we are increasing to accept what do you call that? If you take the medical card, what we call medical insurance, medical card. Yeah, but for these people with medical card insurance, right? Medical insurance. So it depends on how much you pay for your medical insurance. Sometimes medical insurance still cover this type of assay. Okay, so another thing that I want to highlight to you, since you asked there, when you say diagnostics, there are many type of diagnostics. There is diagnostic to detect, and there is also, well, diagnostics for detection to confirm disease, and there is also predictive diagnostics to evaluate benefit of treatment. So this is predictive diagnostics. And then you have prognostic diagnostics, in which you use to evaluate response of treatment or progression of disease. I think all this, who's supposed to provide this definition? Actually, it should be covered under the introduction of diagnostics. I think Dr. Nurul should. Anybody lecture provide you with the definition of this predictive prognostics?
Yeah, it's been covered by Dr. Vanu previously.
Dr. Venu kan? Okay, so I'm glad that it's been covered. But what I mean is that if you search for, panel gene or assay, you will realize that there are several types of assay. Assay for confirming disease, especially in infectious disease. So that is diagnostic merely, basically it's a diagnostic test kit to measure antigen to confirm whether you COVID or not or something like that. And then for cancer, for disease like cancer non-communicable, predictive diagnostics, predictive assay, we use it to assess whether patient will have benefit for that particular drug. So this is what we call predictive assay, predictive diagnostics. And then for prognostic assay or prognostic assay test, we use it to evaluate patient response after taking treatment. Or prognostic, we do the assay to assess whether the disease progress. Let's say you take treatment and then the level of that marker, nothing changed. In fact, it increased more. Then we can say that the patient is progressing to a worse condition or worse situation. So that is prognostic assay. So it depends on whether it's infectious disease or it's a non-communicable disease. Okay, because Kavita was asking about that. But yeah, if you ask me microarray or NGS, yeah, your question, microarray NGS on the research level, yeah, we use microarray NGS. We use it for biomarker discovery. We want to discover new biomarker in disease. We compare it normal with disease, In that sense, we can identify a new marker. But these technologies, microarray, NGS, northern blot, and everything there. And in fact, another amplification-free assay, I'm not sure who's come. The sensor, biosensor technology. Have you heard of it? So that's another technology, amplification-free, in which you can detect whether it's RNA or DNA, depends on the tools. So you use, what do you call that? So you use electrochemical or optical sensor to detect the binding between probe and also your target, or probe or your target. Anyway, I have recorded this session. If you're not satisfied, or to remind you again of what I have said or I mentioned, you can go through the recordings. And if you don't have any more question, I will try to pass you the slide and also the draft A book that I'm going to write to you. Maybe allow me some time, maybe hopefully by Monday. Is that okay?
Yeah, sure.
Okay. So yeah, thank you for your kind attention, student. Okay, before you go, I need to stop this. And I need to have all your names out. And then I will capture your, I need to capture your attendance. Okay. So we have here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10 students. Correct? Yeah, 10 students. So that's for the attendance. Phew. So thank you very much for your kind attention. Have a good night. Have a good rest. Have a good dinner. And have a good weekend, because today is my last day of the week. Tomorrow is my weekend. Have a good weekend. Have a good rest. Have a good dinner. Assalamualaikum and good night, everybody.
Thank you.
Assalamualaikum.
Yeah, welcome. Bye.
Bye, doctor.
See you, doctor.
Yeah.